Usually when we think about a teenager's brain, we picture an abandoned construction zone.
Oh, yeah. It's just a total chaotic mess.
Right, exactly. Things are happening completely out of order, and whoever was supposed to be managing the project has clearly just walked off the job. We kind of casually accept that the delay in higher level thinking, the wild impulsivity, the emotional roller coaster, we accept all of that as a biological bug in the system.
We do. We definitely treat adolescence like this awkward waiting room. There's this pervasive idea out there that the brain is just scrambling to finish building itself so the person can finally start acting like a rational adult.
Like it's just a messy phase you have to survive. But welcome to our deep dive, because today our mission is to explore a truly fascinating 2025 paper published in Nature Neuroscience. And I mean, it completely flips that whole messy teenager narrative right on its head.
It really does. It's a fantastic paper.
The researchers found this hidden timekeeper in the brain that controls exactly when we mature. And more importantly, how our environment physically sculpts our minds during childhood and adolescence.
And, the scale of this analysis is really what makes it so compelling. We're looking at brain scans from 2,676 youth.
That is a massive sample size.
It is, ranging from ages 8 to 23. And the researchers, they didn't just look at one group. They pulled from 3 massive, totally independent data sets. You have the Philadelphia Neurodevelopmental Cohort, the Human Connectome Project in Development, and the Healthy Brain Network.
So we have a mountain of evidence here. And the takeaway that completely blew my mind is that the reason our complex higher-order thinking takes so long to develop, it's not a flaw at all.
Not at all.
It's actually a deliberate feature designed to make us adaptable. And the conductor pacing this decades-long symphony is a tiny structure deep in the center of the brain called the thalamus. But to really understand what the thalamus is doing, we first need to understand the physical roadmap of the brain, right?
Yeah, we have to lay out the geography first.
So the researchers use a framework called the sensorimotor to association axis, or the SA axis for short. Can you explain that?
Sure. So think of the SA axis as like the brain's hierarchy of needs. Development basically always begins at the sensorimotor pole.
Okay, so what exactly is at that pole?
These are the brain areas responsible for basic, really immediate interactions with the world. Things like primary vision, hearing, and basic motor movements.
The survival stuff.
Exactly. The hardware you need just to navigate the physical world safely. But over time, the maturation process slowly sweeps across the brain, moving up this axis until it reaches the other end, which is the association pole.
And that's where the complex stuff happens.
Right. This is the transmodal cortex.
Yeah.
It's the area responsible for higher order cognitive control, abstract decision-making, navigating really complex social hierarchies.
So it's like survival hardware first, complex social software later.
That's a great way to put it.
Because, you need to be able to physically run away from a threat before you need the cognitive ability to negotiate a peace treaty.
Right. The priorities are very clear biologically.
But where does the thalamus fit into all this? Because from what I remember from basic biology classes, the thalamus is usually just called a relay station. Like a biological switchboard operator routing signals from the eyes and ears up to the cortex.
Yeah, and that switchboard operator analogy, that has been the textbook definition for literally decades.
Mm-hmm.
But this paper investigates the thalamus as a master developmental regulator.
So it's not just passively passing messages along.
No, it's actively managing the whole system. The thalamus sends these dense bundles of axons, which are essentially biological wires, we call them white matter tracks. It sends them out to over 200 different regions of the cortex.
200, wow.
Yeah. And the researchers wanted to track how these specific connections mature from childhood all the way into early adulthood.
But doing that in a living human seems incredibly difficult. You can't just open up someone's skull to measure how thick their brain wires are.
It was a massive technical hurdle. So they used diffusion magnetic resonance imaging. To track the wires, they first had to build a highly detailed tractography atlas using adult brain data just to map out the exact pathways.
Like building a master map first.
Right. Then they applied that map to the thousands of youth brains to track a metric called fractional anisotropy, or FA for short.
Okay, let's unpack fractional anisotropy because the mechanics here are fascinating. If we think of those white matter tracks as the brain's fiber optic cables, my understanding is that FA is basically measuring water movement to figure out how thick and well insulated those cables are. Is that right?
That visual works incredibly well, actually. Physically, FA measures the directional coherence of water diffusion.
Okay, so what does that mean in plain English?
Think about it like this. If you drop dye into a still pool of water, It just diffuses equally in every direction, right?
Yeah, it just spreads out like a cloud.
That is isotropic diffusion, which means low FA. But if water is constrained to move along a very strict, highly ordered path-like water forced through a really narrow, well-insulated garden hose, that is anisotropic diffusion.
Got it. So it has a clear direction.
Exactly. That gives you a high FA. So a higher fractional anisotropy indicates tightly packed nerve fibers that are coated in thick myelin insulation, which translates to a highly coherent, incredibly fast connection.
Okay, so let's do a quick recap for you listening. The brain has this SA axis, which is basically its maturation roadmap. It goes from basic survival senses up to complex decision-making. And we are tracking the FA, the thickness and speed of the thalamus's fiber optic cables along that road.
It's a perfect summary.
So here's a question to the listener. Imagine your own brain prioritizing which cables to upgrade first as you grow up. Would you upgrade your survival senses or your complex decision-making first? Because the researchers discovered that the thalamus doesn't use the same type of cables for the whole brain.
Right, the cables literally physically change depending on where they are connecting along that SA axis.
How so?
They found a distinct core matrix gradient. When the thalamus connects to the primary sensory regions, so the survival hardware, it uses cells from its core. These core cells send out dense, highly focused, heavily insulated cables.
The high-speed fiber optics.
Exactly. But when it connects to the association cortex, the complex software areas, it uses matrix cells. And these cells send out sparser, much more diffuse, almost spiderweb-like connections.
Oh, wow. So they are fundamentally built differently.
Yes. Because of the structural difference, FA naturally decreases as you move up the hierarchy.
So the thalamus is sitting in the center, acting as the hub, sending out heavy-duty cables for our survival senses and more diffuse webs for our complex thinking. Which makes me wonder, if your brain is prioritizing which cables to upgrade from age 8 to 23, how does it pace that out?
That brings us to the timeline of maturation. And the results of plotting these trajectories over time were just striking.
What did they find?
The thalamocortical connections do not mature simultaneously, not even close. There is up to an 11-year difference between the earliest maturing connections and the latest maturing ones.
Wait, an 11-year gap in the construction timelines of the exact same brain.
Yes, an 11-year delay.
So if the SA axis starts with survival, the early bloomers must be the primary sensory cables, right? The ones helping us move and see.
Those are precisely the early bloomers. The connections to the primary motor area, specifically known as Area 4, and the primary visual and auditory cortices, they show the smallest age effects.
Meaning they finish early.
Right. They hit their peak connection strengths, their maximum FA, very early in childhood, and then they just plateau. They are built, insulated, and essentially finished.
And the late bloomers, the ones taking a full decade longer, those would be the complex software areas.
Exactly. The connections to the lateral prefrontal cortex, like Area 8BL, and the parietal association cortex, like Area PF. These exhibit the largest and most protracted development.
So they just keep growing.
Yeah, their connection strength keeps climbing, meaning those cables are continually being upgraded and insulated well into a person's early 20s.
And the researchers didn't just map this physically, right? Because I know they cross-referenced this physical timeline with a massive database called Neurosynth.
Yes, Neurosynth is an incredibly powerful tool for this kind of work.
Just for anyone unfamiliar, Neurosynth basically takes thousands of functional MRI studies and links specific brain coordinates to the words used in those studies. So you can see what actual human behaviors are tied to what physical locations in the brain.
And the behavioral mapping perfectly validated the physical timeline they found. The cortical regions, with those early maturing, fast plateauing thalamic connections, they correlate strongly with psychological terms like object recognition, visual perception, and movement.
Basically the fundamental building blocks of interacting with the physical world.
Right. And the regions with the late maturing connections, the ones that are still building in your 20s, they correlate with terms like executive control, decision-making, strategy, and information retrieval.
Okay, so let's pause and synthesize this for anyone listening. Your brain builds its basic physical toolkit incredibly quickly, but it takes over a decade longer to fully install the executive control software.
It's a massive developmental delay.
Think about your own teenage years, Think about the difference between your physical coordination at age 12 versus your ability to regulate your emotions or foresee long-term consequences at age 22. That entire 10-year gap in behavioral maturity is physically mirrored by the 11-year gap in how your thalamus wires your brain.
It perfectly describes the classic teenage behavioral profile. which forces us to ask a much deeper biological question. Why? Why would evolution design a system that intentionally leaves its highest level cognitive centers vulnerable and frankly under construction for an extra decade?
And the answer the paper gives is plasticity. And I think this is honestly the most magical part of the entire system, because we always hear the word plasticity thrown around, but here we actually see the raw mechanics of it.
Right? Plasticity is simply the brain's ability to remain malleable, to physically rewire its connections in response to learning and the environment.
Like wet cement.
Exactly like wet cement. While a brain region's cables are still maturing, while that FA metric is still climbing, that region remains in a plastic state. But once the connection reaches its peak and fully matures, that critical window of developmental plasticity firmly closes. The cement dries.
And the researchers prove that the maturation of these thalamic cables perfectly synchronizes with 3 distinct measurable biological markers of declining plasticity. This isn't just theory. Let's walk through how these work, because it's not just a single switch slipping in the brain, it's an entire chemical and structural shift.
Let's do it.
So the first marker they looked at is the excitation to inhibition ratio, the EI ratio.
Right. The EI ratio is essentially the balance of the brain's accelerator and its brakes. In a young, highly plastic brain, the accelerator is pressed all the way down. There is a lot of excitatory activity, which is usually driven by the neurotransmitter glutamate.
So everything is firing off.
Yeah, it allows neurons to easily fire and wire together. But as the brain matures, inhibitory cells strengthen their outputs to act as the brakes.
And the paper specifically highlights these things called parvalbumin interneurons, acting as those brakes. How do they actually stop the plasticity?
So parvalbumin interneurons, they actually wrap around the main excitatory neurons, and they release GABA, which is an inhibitory neurotransmitter. They act as this stabilizing mesh.
Oh, wow. Literally wrapping around them.
Yes. When they mature, they clamp down on the chaotic, excitable firing, which lowers the overall EI ratio. That stabilization essentially locks the circuit into its final adult form, and that ends the plastic phase.
And what did the data show about the late bloomers?
The study found that cortical regions with the late maturing thalamic connections had significantly smaller age-related declines in their EI ratio.
Meaning those specific prefrontal areas stay highly excitable with the brakes held off for much, much longer. They are primed for learning. Okay, so that's the chemical side. What about the physical structure? Does the insulation we talked about earlier play a role here? Because that was the second marker, intracortical myelin content.
It plays a massive role.
Yeah.
We often just think of myelin as the insulation that makes signals travel faster along the axon, like rubber on a wire.
Right.
But myelin also contains proteins that actively inhibit the growth of new axons. It acts as a literal physical barrier.
Oh, wait, so it's not just speeding things up, it's a cage.
Exactly. Once a circuit is heavily myelinated, neurons can no longer sprout new connections to their neighbors. The researchers found that the areas with the late maturing cables also showed a significantly slower rate of myelin growth.
So the physical cage hasn't been built yet in those association areas? That's incredible. And the third marker they measured was the amplitude of intrinsic BOL-D activity. Now, BOL-D stands for blood oxygen level dependent signal. But I want to make sure I really understand this one. When we talk about intrinsic spontaneous fluctuations, why does a young brain have more background noise than an older one?
Think of spontaneous brain activity, like a massive crowd in a stadium right before a game starts.
Okay.
In a young, highly plastic cortex, the crowd is doing the wave. Large groups of neurons are spontaneously firing together in these huge, high-amplitude waves, exploring different coordinated patterns. It's a very exploratory, flexible state.
Just trying things out.
Right. But as the cortex matures and those inhibitory brakes we discussed take over, the stadium wave stops. The activity shifts into smaller, highly specialized, low-amplitude conversations.
People just talking to the person next to them in the stands.
Perfect, yes.
Yeah.
And the data showed that regions with late-maturing thalamic connections exhibit a much later onset decline in that stadium wave activity. They stay in that exploratory state far longer.
So if we use the analogy of the brain as wet cement again, the myelin insulation and those inhibitory parvalbumin brakes are what cause the cement to dry.
No.
And the thalamus is deliberately keeping the cement wet in our prefrontal cortex.
That is exactly what is happening. By delaying the stabilization of its own inputs to these higher-order areas, the thalamus is acting as the ultimate gatekeeper, forcibly holding the plasticity window open.
So to you listening, think about the implications of this. The longer a brain area takes to develop, the longer it remains physically plastic and moldable. And we can literally measure this by looking at the chemical excitation, the physical myelin, and the amplitude of brain activity. If your brain's highest level cement is still wet in your 20s, what kind of impressions are you letting the world leave on it? Because whatever environment you were in during those years is actively forming the permanent hardware of your adult mind.
Which leads us directly to the environmental impact findings of the study. Because if the cement in our association cortices stays wet the longest, it stands to reason that those specific areas are the most vulnerable and the most adaptable to the environment a person grows up in.
And the researchers tested this, right? They looked at the imprint of the socioeconomic environment on these white matter cables. I know they looked at household factors like caregiver education and family income, but it was the neighborhood level where they found the most profound impact.
Yes. They used factor analysis on geocoded census data to assess neighborhood socioeconomic advantage. And, this isn't just about income. It encompasses community wealth, employment rates, and the general resources available in a zip code.
Right, like a high neighborhood advantage often correlates with safer parks, better community resources, lower levels of chronic environmental stress.
Exactly. And when they cross-referenced that neighborhood data with the brain scans, they found that the higher the neighborhood advantage, the higher the fractional anisotropy in the brain.
So the white matter cables were structurally stronger and better organized in kids from better neighborhoods.
Yes. But the really critical finding is how this mapped onto the SA axis we've been talking about. The environmental effect actually strengthened as you moved up the hierarchy.
Because the primary sensory cables, the survival hardware that gets myelinated and locked down in early childhood, they're no longer plastic enough to be heavily impacted by the neighborhood during adolescence. The cement is already dry.
Precisely. But the latest maturing connections, the ones routing to the association areas handling executive function and emotional regulation, they are highly sensitive to neighborhood conditions.
Because they are the ones left wet and exposed to the environment for the longest time.
Right. So the ongoing stress or support of a neighborhood directly sculpts their final adult structure.
That makes perfect biological sense, but I have a huge point of curiosity here. Did they only look at healthy, neurotypical kids? Because if this core matrix gradient, this 11-year gap, and this prolonged plasticity are fundamental laws of human brain development, shouldn't they apply across the board regardless of mental health?
That is exactly why they brought in the third data set, the Healthy Brain Network, because it is a transdiagnostic clinical sample. Approximately 85% of the youth in this specific data set meet the criteria for a psychiatric diagnosis.
85%. So we are talking about brains that are already facing significant clinical hurdles.
Right. It's a population actively seeking help for various mental health challenges.
So what happened when they ran the tractography and the plasticity markers on this clinical group?
The results replicated perfectly.
Wow. Perfect.
The exact same hierarchical SA axis maturational gradients, the exact same 11-year delay for the association cortex, and the exact same relationships with the three markers of plasticity. It proved that this framework is a universal foundational law of how the human brain wires itself. The hardware rules remain the same, even in the presence of psychiatric conditions.
Okay, so a quick recap before we synthesize all of this. The socioeconomic reality of our zip code literally helps sculpt the physical thickness of our white matter cables, and specifically in the areas responsible for our highest level thinking. If you're listening to this, does it change how you view the importance of community investment? Because things like safe spaces for teenagers, after-school programs, or green spaces, they aren't just sociological nice-to-haves. They're literally the environmental input building the architecture of a teenager's brain.
It really completely shifts the perspective. It moves community support from a purely sociological debate into a conversation about hard, measurable biological reality.
Let's bring all of this together. What this incredibly dense, totally fascinating 2025 Nature Neuroscience paper has shown us is that the thalamus is the brain's master timekeeper. By deliberately pacing out the maturation of our white matter cables, it keeps our higher order association areas plastic for over a decade longer than our sensory areas.
And it achieves this structurally by coordinating the suppression of myelin and holding back the inhibitory interneurons. This protracted plasticity is the evolutionary trade-off that allows humans to adapt to incredibly complex physical, social, and cognitive environments.
And I think the ultimate takeaway here is a message of profound hope. Because the brain's highest level circuitry remains plastic and moldable so deep into our 20s, it means environmental interventions aimed at helping disadvantaged youth can absolutely still be highly effective during adolescence. The window is not closed at age 10.
No, the cement is still wet. Interventions, therapy, and environmental changes in the teenage years aren't just damage control. They are active foundational construction.
But I want to leave you with a final, somewhat provocative thought to mull over. The paper suggests that the way this entire hierarchy kicks off is through sensory feedback. Early sensory experiences, light, sound, physical touch, they tell the thalamus to trigger plasticity in the primary cortices, and then that eventually cascades up the axis to the higher order association areas.
Right, the thalamus is constantly receiving input, just waiting for the right activity motifs to tell it to move to the next stage of development.
So, if the very pacing of our brain development is triggered by sensory feedback, how might the hyperstimulating, algorithm-driven sensory environment of modern screens and smartphones be fundamentally altering the biological timetable of a modern teenager's brain? If we are constantly feeding the thalamus an artificial symphony of light, sound, and immediate dopamine, are we accidentally speeding up or deeply confusing the conductor? It's something we as a society are going to have to figure out. But until then, just remember that the teenage brain isn't broken. It's just a biological masterpiece that takes a very, very long time to paint.